Hi, guys. So I'm going to finish the recording today of my lecture over biological macromolecules. Um, so we ended with proteins, and proteins are composed of amino acid monomers. So remember that the monomers are like the Legos that we're going to build the buildings, which are the proteins with. So amino acids have a central carbon atom that's bonded to that amino group, that acid group, and then they have an R group that varies. And there are 20 amino acids, and each of them has a different R group. You can see from the screen the parts that are shaded in blue are going to be that R group. And that is what makes each one of them unique. So proteins have lots of different functions. Some of them give structural support, like keratin in your hair. There are enzymes that speed up chemical reactions. There's hormones that are your chemical messengers. Actin and myosin are two proteins that help to move your cells and help move your muscles. There are some proteins that transport molecules in blood, antibodies that protect your cells, and then channels that allow substances to cross the membranes. So remember when we talked about lipids and the cell membranes being like the bouncers of the club, not allowing things in and out except for certain chemicals. These channels are like the guys at the back door of the club that if you pay $20, you can get across. So peptides are going to be those joined together amino acids, and they're joined by a peptide bond. So proteins are organized in 4 levels of structure. So there's the primary structure, which is just that linear sequence of amino acids, meaning just like valine, alanine, cytosine, and tryptophan all linked together. Then that secondary structure, either the alpha helix or the beta sheet, is what comes when the primary structure starts to fold on itself. And it looks a little something like this. So you can see that these are the amino acids in this little chain right here, connected by these peptide bonds, and this is the primary structure. The secondary structure is where these amino acids start to bond with each other, okay? Then there's tertiary structure, which is that final three-dimensional shape of a protein. And it's maintained by more of the bonding between those R groups, whether it be covalent, ionic, hydrogen bonding, or even some disulfide bonding, which we didn't talk about. And then there's quaternary structures, and that's basically where you have multiple proteins stuck together. So here's the tertiary structure, where even though we have the alpha helixes and the beta sheets, they're even more linked together. And then the quaternary structure is those polypeptides that are linked together. So that is what the levels of protein organization look like. So why do we care? Well, the final shape of that protein, it determines its function. So if it loses its shape, it loses its function. That's one of the most important things you can know about proteins. Being denatured means that something like that high pH or high heat breaks those bonds, and so it loses its structure and thus its function. Higher levels of organizations of proteins are dependent on that primary structure. So even a single change in one of those amino acids can cause a disease. And one of those diseases is sickle cell anemia. And we'll talk more about that as we get into genetics. So the very final biological molecule that we talk about are nucleic acids. There's 2 types of nucleic acids. One is DNA, and the other is RNA. DNA is what stores all your genetic information in your nucleus, and RNA is what translates it in order to make proteins. So both of them are polymers of nucleotides. So nucleotides, again, are those Legos, and then DNA and RNA are the buildings that they're building. Those nucleotides consist of a phosphate, a pentose sugar, so a 5-carbon sugar, and then a nitrogen-containing base, either adenine, thymine, guanine, or cytosine. There is no thiamine in RNA. It's replaced by uracil. So you can see here that this is just a basic nucleotide structure. It's that 5-carbon sugar with the phosphate and the nitrogen-containing base. This one happens to be cytosine. So here is DNA structure compared with RNA structure, and you can see that the sugars are different. One base is different, that thiamine and uracil, and then the DNA strands are different. DNA has a double strand, while RNA has only a single strand. So they form linear molecules called the strands, and then DNA has two strands, and that leads to what we call semi-conservative replication, which we'll talk about when we get to chapter, I think, 23. So the backbone is alternating phosphates and sugars, and then the bases are projected to one side. And in the DNA, those bases link together with hydrogen bonds so that it forms kind of a helix or a ladder, a crooked ladder. RNA is just single stranded, and it's involved in carrying the information from DNA to make the proteins. So you can see here how the DNA looks like a twisted ladder, and then these blue are going to be your phosphates and your sugars, and then your bonds between um, your adenine and thiamine and cytosine and guanine are going to be what causes the rungs to form, basically. Something important to know is that adenine only binds with thiamine and cytosine only bonds with guanine in DNA. Now, in RNA, uracil is going to take the place of thiamine, but A only bonds with T and C only bonds with G. And you can see here it's because of the number of bonds. Adenine can only make two bonds, and cytosine and guanine can make three bonds. There is one more important molecule, adenosine triphosphate, and it is the high energy molecule of the cell. So you've all heard that mitochondria is the powerhouse of the cell. Well, the reason it is because it produces ATP. And so we talked about bonds have energy in them when they're broken or put together. It either takes energy or releases energy. And so when we break the phosphate bond in ATP, then energy is released. So this is a picture of ATP. You can see the three phosphates, which is where it gets the tri name. So this is adenosine and this is triphosphate. And the last two phosphate bonds are unstable and they're easily broken. So when we break this bond right here, it releases energy. And that energy can be used to do things in the cell. For example, it can be used to move your muscles or to send signals, things like that. So that is the end of this lecture. If you have any questions, please let me know. And I will see you on Monday.